Hi everyone, welcome to this lecture on the cardiovascular system. We've just come from talking about the heart, and now we expand our horizons to the system as a whole, which involves all of the vessels in the body. There's a variety of different vessels and they have different properties to them, so we'll talk about those in this lecture, and then next time we meet we will continue talking about some of the physics and mechanics involved in how the heart can push fluid through this network of tubing. Okay, so the vessels, in a real general sense, there's a difference between arteries and veins. There's different kinds of arteries and different kinds of veins, but generally speaking, they have some of these contrasting properties. This shows a slide of a vein and an artery, the cross section of them. And a couple things I'll point out here is that you'll note that the artery is a little bit more round in shape, whereas the vein is a little bit more irregular. Okay, this is due for a variety of reasons. One, the artery is going to have a much thicker, what's called tunica media, this part right here in the middle that has all the smooth muscle cells. Arteries also have a lot more elasticity to them, specifically the larger arteries. They have an elastic membrane in their inner layer and another elastic membrane in the outer layer. The elastic membrane is basically just a network of lots of elastic fibers. And that's important because arteries are high pressure vessels. They endure a lot more pressure because of their proximity to the heart. The closer a vessel is to the heart, the more pressure it's going to absorb because it's really the beating of the heart that's pushing the pressure of blood into the system. Veins are a little bit more wide open. They have a bigger lumen actually, and this is going to help allow a little bit more blood flow, despite the fact that once blood reaches the venous side of the body, it's more of a low pressure system. So that's generally kind of how the appearance of these look. Regardless if we're talking about a vein or an artery, they're all going to share three basic layers. There's the innermost layer, which is this one, and this is called the tunica intima, meaning it's most closely associated with the blood, which would be passing through the middle of the vessel. By the way, the middle of the vessel is given this name, the lumen. So if you ever see this word lumen, it just simply means the inside of a vessel. Okay. Well, the layer that's directly exposed to the lumen is called the tunica intima, and it's this first layer here. Tunica comes from the Latin word for tunic, which is still a word we use today, but if you're unfamiliar with that term, it's basically like a cloak or a garment of clothes, and so whatever anatomist named these thought that these were like different clothes that are sequentially put on the vessel. So you have the tunica intima. The next one is the tunica media. That's the one that is going to be in between the outer and inner layer of the vessel, and this is where the smooth muscle is found. This is an important layer because it's the contraction and relaxation of all these little smooth muscle cells which are going to change the diameter of the vessel, either artery or vein. When these smooth muscle cells contract, they all pull in with one another, and that's going to actually shrink down the vessel. This is called vasoconstriction, and it's going to be a very important process in regulating blood flow. In contrast to that, when these smooth muscle cells relax, the vessel can kind of pull away a little bit, and it is going to open it up. This is called vasodilation, and that is going to make it much easier for blood to pass through. So kind of two tools here that the cardiovascular system can employ to either increase or decrease blood flow just simply by contracting or relaxing the muscles that surround the vessel lumen in the tunica media. Now, the way that this works is that there's little nerves that will pass into here, and then there's kind of a network of nerves, smaller nerves inside the tunica media, so that if a signal comes from the brain and it reaches the tunica media, it will spread out and cause all these muscle cells to either contract or relax. And this is happening in all the vessels of your body, all over your body, so it's pretty fascinating when you think about the control that your brain has on this vast array of tubing that's carrying blood throughout your body. Another thing to note about arteries is that, well, both veins and arteries, is that at a baseline moment, so at any given moment when nothing really exciting is happening, they have this kind of baseline contraction. This is called the tone that they have, or specifically vascular tone. Vascular tone refers to how contracted the muscles are. And so generally arteries and veins will be slightly contracted, and then they can even contract further, or then they can relax. This is partly seen here in the rippling of the tunica intima here. You see how it's kind of a pleated, rippled shape? That's because it's kind of scrunched up a little bit. What's kind of also interesting is that while there is blood passing through the artery and there's blood passing through the vein, typically there's kind of a lot of connective tissue here in the tunica intima, which doesn't really allow blood to get from the vessel and from the lumen of the vessel into the tissue surrounding the vessel. And so arteries themselves and veins themselves will have little blood vessels running in and out of them. These are called the vasa vasorum, which literally just means the vessels of vessels, small vessels of vessels. So a blood vessel has little small blood vessels running in and out of it to give itself the nutrients that it needs to work. So kind of several tiers of vessels going on. Okay, here's another kind of look, a cartoon look to make it a little bit easier to see. Here we see our three layers, the three tunics, tunica intima, tunica media, tunica externa. And here we're comparing an artery with a vein. You're going to see that the vein also has the three layers, and so do all other vessels. There's different types of arteries, different types of veins, but regardless, they're all going to have those three layers. One of the things that's going to make an artery unique, though, like we said, is it's going to have a thicker tunica media. And then the other thing is that it's got a lot more elasticity to it. Okay, one of the things that elastic arteries have is this band of elastin fibers that are embedded in the tunica intima. This is called the internal elastic membrane. It's found in arteries. It's not found in veins. And then also some of the pretty big arteries, not all of the arteries, but a lot of the bigger ones will, in addition to the internal elastic membrane, will have an external elastic membrane, and that is going to be kind of a web of elastin fibers that are found in the tunica externa. The endothelium is going to be the innermost lining of cells, and again, this is usually just a single layer of cells that are going to be found facing inward, and they generally are embedded in kind of a collagenous matrix, an extracellular matrix, and then there's usually some connective tissue that is they're anchored on called the basement membrane. But there are little cells that live in the lumen of these vessels, and then also there are these little, like we said, maybe the blood that's passing through the vessel can give nutrients to these little cells that are facing the innermost part of the lumen, but all of these cells back here, they don't really get the nutrients passing from the blood because of all of the connective tissue found in the tunica intima. So to remedy this problem, there are vessels that are actually inside the vessel. And so you can see this is a cross-section of what we would call a vasa vasorum, which is going to be perfusing and sending nutrients to the tunica media and the various fibroblasts and different cells that are in the tunica externa. And then in addition, there are these little nerves, little small nerves running along the vessel, which we call the nervi vasorum. And the nervi vasorum are going to be the things that tell the tunica media to either contract or relax. You see a lot of the same stuff in the vein, but just note that there is no internal elastic membrane. There is no external elastic membrane, and the smooth muscle, again, is quite a bit more thin. The reason for all of these features is the fact that veins are low pressure, arteries are high pressure, so in order to sustain that chronic high pressure, there's got to be a little extra structural support and strength in the arteries. Here's kind of a nice summary table that's found in the book about the difference between arteries and veins. This would be kind of a good table to study. You could cover these up and see if you can recall what the difference is between the tunica intima of arteries and veins, tunica media of arteries and veins, and the tunica externa between arteries and veins, as well as their general appearance. All right, now you can subdivide the arteries and veins into different classes depending on their size. So here's a nice figure that came from a different book that shows some of the different vessel types. There are, and it all really depends on the vessel's proximity to the heart. So closest to the heart, right off of the heart where the aorta is, this would be what we would call an elastic artery. An elastic artery simply because there's a lot more elastin fibers in this vessel than there are in the other different types of arteries and veins. This is important because every time the left ventricle, which is one of the higher pressure chambers in the heart, which really does most of the work, and anytime this ejects a bolus of blood, there's going to be a bolus of blood is basically just the large amount of blood that leaves the ventricle each time the heart contracts and it ejects the blood out. This kind of conglomeration of blood is going to kind of put a lot of pressure on the vessels because it could go from like 80 millimeters of mercury all the way up to 100 millimeters of mercury. That's a pretty big swing in pressure. And the vessels here have a lot of compliance, meaning they can stretch to accommodate that extra increase in pressure so that it's not so detrimental to the tissue. So that's why it's important to have elastic arteries. If these were really rigid vessels, every time that increased pressure came into the heart, it would put a lot of strain on the vessel. It could perhaps maybe rupture the vessel or tear the vessel. So the elasticity helps dampen that pressure spike. Okay, as we move further away from the heart, basically the aorta, which is the big elastic artery, is going to branch, and then there's branches that come off of it, and then there's branches that come off of those branches, and then there's branches that come off of those branches, and then the vessels, as they get further away from the heart, they don't need as much elastic connective tissue, and so the proportion of the tunica media to the rest of the vessel is actually getting a little bit larger, and so we call these muscular arteries because they no longer have as much connective tissue and elastic tissue. Most of it is really just the tunica media. There isn't really that whole lot to the tunica externa or the tunica intima. The division between an elastic artery and a muscular artery is not, black and white. We kind of have a general idea of what an elastic artery is and a general idea of where a muscular artery is, but where an elastic artery stops being an elastic artery and then starts becoming a muscular artery is a little bit ambiguous. Muscular arteries will then go through and perfuse a lot of different parts of your body, and then muscular arteries will then branch into a bunch of smaller vessels, which are called the arterioles. The arterioles are the smallest of all of the arteries, and the size of these is about 30 micrometers or less. And just to give you some kind of sense of the scale, 30 micrometers, an average red blood cell is between 6 and 8 micrometers, so these vessels are big enough to allow maybe 4 or 5 vessels to fit at one time through a cross section of the vessel. Once the arterial there'll be arterioles, and then the arterioles will branch, and then they'll continue branching, and then eventually we'll arrive to what are called terminal arterioles. And the terminal arterioles will then each individually perfuse what's called the capillary bed, which is kind of this place where it's actually a super important place physiologically speaking, because this is the place where you, in essence, get all of the nutrients that you'll ever need and get rid of all of the waste products. Okay, so here we see a picture of a terminal arteriole, which is then leading to this network of tubes called the capillary bed. Well, the capillary bed is going to be a place where all of your cells, for the most part, live. And any cell that will ever be perfused, meaning any cell that will ever receive nutrients from the blood, exists in a capillary bed. The reason being that nutrients can't really get out of any of these previously mentioned vessels because of all of the tissue. But once we get to the level of a capillary, the capillary tube is so small, the wall of it is so thin that then it allows nutrients to leave the blood and bathe all of the cells that are living in it. We'll talk at great length here about the capillary bed later. It's just, like I said, a super important place to discuss when we're talking about physiology of the body. Now this is where the big division happens, though. Once we've reached the capillary bed, we now consider the blood to not be coming away from the heart, and now we consider the blood to be coming toward the heart. So this is where the division between the arteries and veins happens. So in essence, capillaries are neither a vein nor an artery. They're a kind of interesting tube network that exists between the arterial and the venous side. So now as blood passes through the distal end of the capillary bed, now we're on the veins side. And then we go from a smallest vein to a little bit of a bigger vein to the biggest vein, and then we make our journey back to the heart. The smallest vein is called a venule. Then we get up to what's called a medium-sized vein, and all of the medium-sized veins will then pour their contents into what we would call a large vein. This would be something like the superior or the inferior vena cava, and then that gets back to the heart. One of the really interesting things about medium-sized veins is that they have valves. That's not found in any of the arteries. What happens is that the endothelium, that inner lining of cells, it will kind of make these little infoldings to create valves, which are going to help the blood flow back to the heart. We have in the veins, we have a low pressure system, and so it's harder for blood to get back to the heart without these valves. Okay, so here's kind of a cross-sectional view of some of these vessels. This is just straight from the book, and you can kind of start to see the different comparisons here. The elastic artery over here, it's got lots of elastin fibers in it. Pretty substantial tunica intima, a tunica externa, and then once we get here, we lose a lot of some of that elastin fibers. It's mostly just the meat, mostly just the tunica media. And then once we get to the arterial is really, really small. It's really only just a couple cells thick, the tunica media. And then the tunica externa is very, very, very thin. And then the venule looks pretty comparable to the arterial. The only difference is, look at the tunica media. There's hardly even one cell in there, and whereas the arterial is quite a bit more muscular. So venules are probably more like capillaries in that sense. Once we get to the medium-sized veins, here's where we start to see the appearance of valves. And this isn't found in any of the other vessels, and this is a kind of a cross-sectional view of what a valve looks like. Basically, blood will be pushed through the valve, and then once gravity is pushing it backwards, it shuts the valve so that it can't flow backwards. So blood is continually moving forwards, it's not able to move backwards. This figure shows how blood is partitioned through the cardiovascular system. In essence, there are two circulations. There's the one that brings blood from the heart into the body, which I've sort of alluded to earlier, but all the blood that's leaving from the left ventricle of the heart and then returning through the right atrium of the heart, this forms what's called the systemic circulation. It contains 84% of your blood. And all the vessels that are bringing blood from your heart to the various cells in your body, these are called the systemic arteries. Should be right here. And all of the vessels that are collecting the blood from the cells in your body and bringing it back to the right atrium, these would be called the systemic veins. And they will hold about 64% of all of your blood. So a majority of your blood is right in all of the vessels collecting blood from most of your body. Then kind of the unique circulation would be the one that goes to the lungs and back, and I know this convention isn't totally accurate because the lungs is still technically considered part of your body, but you know what I mean. This would be your body, everything except your lungs, and then this would be your lungs. This circuit involves the right ventricle, blood leaving the right ventricle, and then going to the lungs, and then the blood leaving the lungs coming back into the left atrium. So that would be what we call the pulmonary circulation. Vessels bringing blood back to the heart would be the pulmonary veins. Blood leaving the heart and going to the lungs would be called the pulmonary arteries. And so you can kind of see how all the blood is arranged and divvied out into your cardiovascular system. The one thing I want you to note here, you don't have to know all the specific percentages of how blood is passing through the different veins in your body and arteries in your body. But I do want to bring up the fact that there's a majority of the blood is in your veins. This is important What happens is that you have a lot of high pressure coming off of the left ventricle, and by the time that pressure, that blood is driven throughout all of your body and dispersed through all of the networks of capillaries and everything, it loses a lot of the pressure. So the veins that are carrying blood back to your heart, they have a low pressure, and it's a little bit harder for them then to push blood back to the heart. So it ends up just kind of pooling in the vessels here. The blood gets kind of stuck in a way here. Now it's continually moving through, but it bulges and distends the vessels so that it kind of pools in the veins here, which is why a majority of it is here. And the arteries, they're really constricted. They've got muscle around them. They don't really distend out that much, where the veins, they do. So they're these nice reservoirs of blood. And what can happen is, in an event of severe blood loss, what the body can do is this thing called venoconstriction. This is not to be confused with vasoconstriction. Note the E here, venoconstriction. This is particular to the systemic veins, and what it is it's a special neural signal that gets sent out to the veins, causes them to constrict, and this is going to push some of that blood that's being stored in the veins back into the systemic arteries. If these vessels here constrict, they push the blood back into the heart, and then it's now again into the arterial system. This is good. It helps keep the pressure in the arteries high, and that's vital. If the pressure in the arteries falls, it's probably that there's not going to be enough pressure required to perfuse, or that is to send blood to the tissues of the brain, and that's a really important organ to keep going. So interesting compensatory here response for the veins. All right, now let's talk about these interesting tubes, the capillaries. Neither an artery nor a vein. Capillaries are found in these regions called capillary beds, as we maybe mentioned earlier. The capillary bed is a place where all of the cells live, and there's a whole lot of interesting dynamics here happening, which we'll talk about later. But for now, I want to just focus on the tubes themselves. There are a whole lot of capillaries in your body. In fact, pretty much your entire body really is cells and capillaries. If you ever seen the exhibit Body Worlds, it's a pretty cool exhibit for those interested in anatomy. What it is people that have donated their body to this organization will have their different organ systems highlighted. The way they do this is they plasticize the different organs, and then they dissolve the rest of the body, so you can really see organ systems in their kind of unique state. So this is one that they've done of the cardiovascular system, and you can see a lot of the vessels here. Like this would probably be a muscular artery, and then a lot of these smaller tubes here are probably arterioles, and then in all of these empty spaces is probably where all of the capillary beds would be, but the capillaries are so small they probably didn't get preserved here. But just a lot of interesting specimens, and you can just see how much tubing there is in your body. I mean, your body is full of tubing, and they even have other organisms like a shark, and you can just see how densely packed the vasculature is in an organism like this. Well, most of that is capillaries. Again, what you're seeing here isn't capillaries. The capillaries probably were dissolved, but even still, for every length of this tubing, there's probably 3 or 4 or 5 or 6 times more tubes that are capillaries. So there's a lot of capillaries in your body, is I guess what I'm trying to say. There's really no cell in your body that's going to be more than 100 or 200 microns away at any given time from a capillary. So they're everywhere in your body. There's 3 types of capillaries. There's the continuous capillaries, the fenestrated capillaries, and the sinusoidal capillaries. And they vary in the degree to which they are sealed, so to speak. So a continuous capillary, it has very tight junctions between the endothelial cells. Each one of these little things here is an individual cell. They're forming the tube of the capillary. And in a continuous capillary, these are very tight seals. They don't allow the passageway of much. Okay, generally a tight junction like this would not allow anything to pass through, but there are these things called intercellular clefts, just this slight little infolding here at the junction that will allow some stuff to pass through. things that can be exchanged through the walls of a continuous capillary. It's really mostly small stuff. So things like monosaccharides or disaccharides, simple sugars like glucose or fructose, those types of things. Different amino acids, probably little or smaller vitamins, and different electrolytes. Things like electrolytes would be ions, like potassium ions, chloride ions, calcium ions, sodium ions, and also different gases like CO2 and oxygen. Also some of the smaller hormones. The things that are not exchanged through these types of capillary walls would be larger proteins. Okay, there might be some peptides that can make their way through these intercellular clefts, but not like a protein, not something that's made of more than 10, 20, or 30 amino acids. Also, then, if proteins aren't allowed, then certainly organelles and anything bigger than a protein, like a cell, could not be exchanged. There is one exception here when I want to talk about first cells. This sometimes throws students off. Some cells, specifically leukocytes, mostly the neutrophils, a certain kind of white blood cell, the neutrophil, which we talked about earlier, they are specially trained to wiggle their way through these cell junctions. So they are kind of an exception, but normally cells are not able to pass through here. And just to give you kind of a size, a sense of scale, the diameter here of a capillary tube is probably 8 to 10 microns thick, and for reference, a typical erythrocyte or a red blood cell would be about 8 microns thick. So you can imagine that this tube is just big enough to let one red blood cell through. And generally, red blood cells have to move single file and even have to maybe even squish and bend a little bit to fit through a capillary tube. All right, so that is the continuous capillary. One last note I guess I forgot to mention is that because they don't exchange nutrients as rapidly as some of these other ones, these types of tubes are generally well suited for cellular transport. So they have transport vesicles, meaning they can get rid of stuff through endocytosis, or they can uptake stuff through, or they can get rid of stuff through exocytosis and obtain things through endocytosis, and then they can process the nutrients and then maybe export them or import them to the neighboring cells. So well suited for cellular transport. These types of capillaries are found everywhere. They're the most abundant. The next type would be the fenestrated capillaries. Fenestrated is named because of the word fenestra, which is Latin for window. And so they have these little windows in them, or these little pores. These are going to increase the permeability and the rate of exchange and the types of things that can be exchanged through the walls of the tube. And basically these ones are going to allow the exchange of all this stuff which the continuous capillary walls could exchange, but then in addition to that, these will also allow the exchange of proteins, and probably more smaller proteins. This is the type of capillary that's found in places where there needs to be great absorption occurring in the body. So for example, this would be the small intestine. The small intestine is the place where most of the nutrients in your body are absorbed. So there needs to be a lot of holes through which things can pass. Additionally, you would find fenestrated capillaries in the kidneys, mostly because the kidneys are a place where a lot of metabolic waste products and things like that are cleared from the blood, so they have to be able to get out of the blood. You also find these types of capillaries where there's in different endocrine organs. Endocrine organs are things that make hormones. Hormones are basically a variety of different proteins or lipid or other different types of things that are made and do stuff in your body. They turn stuff on or turn stuff off. Well, the endocrine organs are going to be making these things, and they have to get into the blood pretty rapidly. And so there's fenestrated capillaries found in these places. A couple of examples would be like the hypothalamus and the pituitary gland of the brain. Also the pineal gland of the brain. These are some endocrine organs that you find in the brain that help regulate everything. And And if there's a rapid endocrine response that needs to happen, the cells in these places will make their contents. Those contents will diffuse into the bloodstream and then be carried throughout the body to exert their effects. The last type would be, it's more of the rarer type, the sinusoidal capillaries. These are found in the liver, they're found in the spleen, they're found in the lymph nodes, they're also found in the bone marrow. Basically, these are found in places that have high cellular traffic. So for example, the spleen is a place where a whole bunch of red blood cells and white blood cells are mingling. They're bumping into each other, moving around. So the spleen is a really kind of spacious organ. It has a lot of cavities and for places for cells to occupy and move around. And so there's a lot of blood stuff coming out of the blood and going into the blood in the spleen. So this allows things like cells and things to get into and out of the blood, because the sinusoidal capillaries have very large holes in them. These are called intracellular gaps. So they're kind of the most permeable of all three types. And then similarly, the lymph nodes, this is a place where white blood cells are continually bringing pathogens and other things. So it's a high traffic area where stuff can get into and out of the organ if need be. Same with the bone marrow. Bone marrow is where a lot of cells are made, so they have to get from their production site out into the bloodstream, and the way that they do it is they jump right on into the sinusoidal capillary. And then the liver is a place where a lot of plasma proteins are made. We talked about these different plasma proteins in the blood chapter, so you could review that if you're not sure what I'm referring to, but it's things like albumin or different transport globulins, things like that. And imagine this sinusoidal capillary being one that is found in the liver. All of these cells then surrounding it would be little liver cells, and the liver cells are always making these different plasma proteins, and the plasma proteins that are made will then find their way through these holes, and they are able to rapidly get into the bloodstream because of the permeability of these types of capillaries, the sinusoidal capillaries. All right, now on to the capillary bed. This is a unique place where a lot of different stuff can happen. This, in essence, is where a lot of physiology occurs. This is the place of great exchange, all right? This is the place that all of your cells live. And so if you think about your body, the analogy I like to use here is if you think about your body as like a really big city, let's say the Twin Cities. And in the same way that here in the Twin Cities we've got, we've got I-94 and I-35 and 694 and 494, these major freeways to get around rapidly, and then you might exit a freeway onto a highway or a more busy road, and then eventually from that busy road you would make your way to a smaller, quieter residential street where all the houses would be found. Well, in that kind of analogy, your body is like a collection of these little houses that live in these little neighborhoods, which are called the capillary bed. And nutrients are brought to these cells through the capillaries, and then they will enter the capillary bed and then give the cells what they need, and then the cells will release their contents into the blood, and there's this really great exchange here that happens which keeps you alive. Well, the blood flowing through the capillary bed has to be extraordinarily regulated for a variety of reasons. One, if a cell is deprived of blood flow for too long, it will die. And the caveat here is that there's way more capillary tubes in your body than you have enough blood for. In fact, the book talks about if all of the blood in your body was in the capillaries, there would be none left to be in the arterioles or the veins or the arteries or the heart. Okay, so there's just far too much tubing than there is blood to go around. So your body has a sneaky way of regulating its blood flow. It never perfuses all capillary beds in your body at once. It at times perfuses one capillary bed and then shuts off, and then perfuses a different capillary bed and then shuts off. And so there is a really intense regulation of blood flow that happens at the level of capillary bed. By far the biggest way that blood is regulated is by opening and closing the arterioles. Arterioles are kind of the passageway into the capillary bed. And so if blood flow needs to be reallocated or brought to a different part of the body, it will shut off the capillary beds that are doing okay, the capillary beds that have cells that have been well perfused, so that it can send that blood elsewhere and not waste its time perfusing a place where cells are already okay and well provided for. So that's the biggest way that blood flow is regulated. The second way that it's regulated is there are these things called anastomoses. An anastomose is a general term that refers to any place, anytime you have a vessel connecting two other vessels. And here, because it's a vessel that branches between the arterial and the venous side, this is kind of the division again between the smallest artery and the smallest vein, we call it an arteriovenous anastomose. This can open and allow blood to bypass the capillary bed, but it can also shut. It's got all these bundles of smooth muscle rings surrounding it, and they can contract and shut this off. If it shuts off, then blood will not pass through and it will continue on, or it will get forced through the capillary bed. So here's kind of a second place where blood flow through the capillary bed is regulated. The first is kind of the incoming arterial, the second is the anastomose, and then blood that does go through into the capillary bed, it will pass into this kind of, it's a little bit of an interesting structure because it's half of an arterial, but it's now in the capillary bed, so it's not totally an arterial anymore. We call this structure a metarterial, basically the arterial that allows fluid to come through the capillary bed. Now, a third way that blood is going to be regulated, blood flow is regulated through the capillary bed, is through the opening and closing of these things called precapillary sphincters. Precapillary just means the structure before the capillary, and a sphincter is any kind of ring of muscle that can open or close a tube. And so here we have these rings of muscle, and they'll contract to squeeze this shut so that no blood can pass through, and then they could at another moment open and allow blood to flow through. So you've got super intense control on a tube capillary level, capillary by capillary. At one point this one might be perfused and then shut off, and then at another moment this one might be perfused and then shut off, and then maybe this one will be perfused and then shut off. So really intense control of how blood is flowing through the different parts and regions of your body. Most of this is controlled through a local regulation mechanism. Local means that it's happening at the level of the cells. So generally how this works is that there'll be cells here living in these little neighborhoods, and let's say this region of cells is hungry, so to speak. It needs nutrients. They need nutrients. They'll release a variety of factors, different chemicals, which will act on the smooth muscle to either open or close them. So if they need something, they'll release these factors which open them and allow it to perfuse. And if they're satisfied, then they probably won't be releasing those factors, and then this tube would shut closed again. So it all happens at the level of the cells. This would be different than like a systemic response. A systemic response means that it's a response that's done throughout the entire body and controlled by the brain. But the regulation of blood flow happening here is not controlled necessarily by the brain. It's really just happening right here in its little local spot. So that's nice because it alleviates the burden of the brain to figure out what parts of the body needs to be perfused or not perfused. Now, the way that blood flows through the capillary bed is given a special name. It's called vasomotion. And vasomotion just basically refers to that discontinuous fashion of perfusion that I've alluded to. At 1 minute, one capillary will be perfused and then shut off. At another minute, another one will be perfused and then shut off. And then another another one will be perfused and then shut off. And I have kind of a little video here to show you a little bit of what this looks like. This isn't specifically precapillary sphincter and a capillary. This is actually an arterial. But you know how we said earlier where an arterial can shut off and then open up. So this kind of shows you a little bit of a picture of that. Vasomotion refers to the spurting of blood through the capillary tubes. So it's not really technically what you're seeing here in this video, but it's a similar process. So here you're seeing the walls of an arterial. It's open now, and you can see all of the many red blood cells rushing through. They're perfusing the area of cells that need nutrients, and then once those cells are satisfied, the vasodilatory factors are going to cease, and then this wall will then kind of shut and stop blood flow. And then when when the cells need blood flow again, they'll release those vasodilatory factors, the things that cause vasodilation. The blood will rush through because the smooth muscle is now relaxed. And then once they're okay, then they stop releasing those factors and the smooth muscle will then close shut again and occlude blood flow. And then once they need blood flow again, it opens up and then they pass through and then it shuts again, and it just continues on through this process. This would be something like probably a really, really small artery, and then this would be the arteriole that is now, blood is entering, and then maybe down here would be a capillary bed. I always love watching these. It's so interesting to just think about how many cells are just getting packed and rushing through, and you see these little tiny blood cells rushing through there. It's just fascinating to me, all the activity that's happening in the body. All right, we will pause here, and when we pick it up next time, we'll then start talking about the specific names of different arteries and veins in the body.